So, very good morning, dear students. So, today we'll begin with the new topic, new chapter, biomolecules. So, in the previous class, we had completed the cell cycle and cell division, isn't it? So, we are going to commence with this biomolecules. You should remember that biomolecules is nothing but biological molecules, where we are going to study about macromolecules as well as the micromolecules, which play an important role in living organisms. So, we'll be discussing in this chapter concepts about carbohydrates, types of carbohydrates, proteins, types of proteins, lipids, and types of lipids. Apart from that, nucleic acid also will be studying about the DNA and RNA. So the types of nitrogenous bases that they have, and if they are having nucleotides, what we call them as, and if they are having nucleosides, what are the terms that we utilize? All this we are going to study about. So this biological molecules, knowledge of it will help you to understand the living organisms better. And this is more concerned with biochemistry, that is, the chemical molecules which are associated with living organisms. So, the chemistry part of it is also what you have to understand about. So, the biochemistry and so we are going to study here about the biological molecules. How do we extract them, and what are the different process of trying to analyze what type of molecules are all present in the living organism. So this is from the viewpoint, it might be a dry chapter for most of you because it is more of chemistry and a little of biology. Okay, so we will be trying to understand those, the structures and other things. You can just understand them, and during examination they might not ask you the structural formula. More of questions on chemistry would be there, biology would be there, and you would also be studying in this topic enzymes, types of enzymes, the activity of enzyme, the mechanism of enzyme, how they are going to act on the substrate. All that we are going to study in this chapter. So it's a interesting chapter, uh, and it also requires your alertness throughout this topic, because there would be chemistry part of it, and you would also have to understand on those lines. So let us begin with this chapter, the biological molecules. Excuse me. So the biomolecules are chemical compounds found in living organisms. So biomolecules is an almost an acronym of biological molecules. So the definition of biomolecules are the chemical compounds which are found in living organisms. We call it as biomolecules. So the chemical compounds can be organic and inorganic compounds. Okay, so it can be in the form of ketones, aldehydes, carbohydrates. So the organic compounds, the inorganic compounds like HCl is found in the secreted in the stomach. Okay, so there are many inorganic compounds which are also present within the living organism. So we are going to study both of them. So we are going to study the organic and inorganic compounds present in the living organisms. So biomolecules are chemical compounds found in living organisms. So these chemical compounds include organic and inorganic compounds. So here you can see in the backdrop a lot of this organic and inorganic compounds. So you can see the uh, amino acids, R amino acids, RCH, uh, NH2COOH, the amino acid groups, amino groups. Then you can see the carbon complex organisms like carbohydrates. Okay, so the ribose nucleic acid, sugars, and phosphate group. So the DNA. So these are all the biomolecules that we would be studying in this chapter. So that is what those slides indicate. How do we analyze the chemical composition in the tissue? So to analyze a chemical compound in a composition in a tissue, what we are going to do is grind a living tissue. It can be a vegetable or piece of liver, etc. So it can be leaves. Here they have taken in the slides, you can see the leaves. So in a mortar and pestle, they are going to grind it. Okay. So grind a living tissue, which can be a vegetable or a piece of liver, etc., in trichloroacetic acid. This trichloroacetic acid is CCl3COOH. Okay. Uh, so to get a thick slurry, so you get a thick slurry. So you have to grind this living tissue in trichloroacetic acid. So Cl3CCOH OH to get a thick slurry. So once we have obtained this thick slurry of this living organisms dissolved in trichloroacetic acid, we strain, we filter or strain it through a cheesecloth or cotton to get two fractions. So such as filtrate and the retentate. So the filtrate would be the acid soluble pool that you'll be collecting in this bowl, whereas the retentate, the acid insoluble fractions will be present above. So the trichloroacetic acid soluble pool will be the filtrate that you have collected in the bowl, and the retentate is retained on the cheesecloth or cotton, so which is acid insoluble fraction. So you have acid soluble fraction in the bowl, acid insoluble fraction or the retentate is noticed above the cheesecloth or cotton. Okay, so the filtrate and retentate you have. So filtrate has this acid soluble pool. Now in the filtrate acid soluble pool, they contain biomolecules, biomicromolecules, so whose molecular weight is less than 1000 Dalton. So we find that they contain biomicromolecules whose molecular weight is less than 1000 Daltons. And we find the retentate, that is the acid insoluble fraction, which contains biomacromolecules whose molecular weight is greater than 1000 Dalton. Okay, so we are going to see the retentate or the acid insoluble fraction, which contains biomacromolecules whose molecular weight is greater than 1000 Dalton. So the filtrate will have biomicromolecules whose molecular weight is less than 1000 Daltons, and we have the retentate, they'll have this biomacromolecules whose molecular weight is greater than 1000 Dalton. So that is how we are getting the filtrate and retentate within this the mortar and pestle when they are filtered through a cheesecloth or cotton. We get this two fractions, filtrate and retentate. The filtrate, which is collected in the bowl, they would be the acid soluble pool, and the retentate, which is found on the cheesecloth, they would be the acid insoluble fraction. They have the biological macromolecules. So the filtrate has biomicromolecules whose molecular weight is less than 1000 Dalton. So the retentate will be having a molecular weight which is greater than 1000 Dalton. So now the analysis of inorganic compounds. So to analyze, the previous slide is analysis of chemical composition in a tissue. So now in particular, if we are analyzing the inorganic compounds, we weigh a living tissue, wet weight, and it is dried. So they are dried to evaporate the uh, water. So the living tissue, wet weight is weighed, and they are dried, dry weight to evaporate water. Okay, so we weigh a living tissue, which is the wet weight, and it is dried. So the dry weight is also there. Weigh a living tissue, and it is dried to evaporate water. Once it is evaporated to water, it is fully burned to oxidize all carbon compounds to gaseous forms. So we remove all the organic compounds by this method. So the dry weight of that material, living tissue material, it is burned to oxidize all the carbon compounds to gaseous form. So carbon dioxide and water vapor is removed in the gaseous form. So the burned part of this dry weight of the living tissue, it forms the ash. So this ash contains inorganic elements which we can analyze. So they have inorganic elements like calcium, magnesium, sodium, potassium, and inorganic compounds they would be having like sulfate, phosphate, sodium chloride, calcium carbonate, etc. So to analyze the inorganic compound, what we are doing is we weigh the living tissue, the wet weight, and it is dried. So the dry weight is there. So and it is dried to evaporate water. So this dried weight of the living tissue sample, it is burnt. Why do we burn it? Is to evaporate all the carbon compounds into its gaseous forms like carbon dioxide and water vapor. We remove the organic compounds of it. So it is fully burnt to oxidize all carbon compounds to gaseous form like carbon dioxide and water vapor. So the remnant of after burning, we call it as ash. It forms the ash. So this ash contains inorganic elements. It would be containing. When we analyze them, we can find calcium, magnesium, sodium, potassium, etc. And they will also have inorganic compounds, not only inorganic elements like calcium, magnesium, sodium, and potassium, but they also have inorganic compounds like sulfate, phosphate, sodium chloride, calcium carbonate. So these are all the inorganic compounds that we also notice in this ash. So when we compare the elements which are found in the non-living and living matter, this is what we notice. So when we compare the elements in non-living and living matter, so the following things are noticed. So we have this percentage weight. Elements that we have is hydrogen, carbon, oxygen. So these are the elements. So in one column, we have listed out the elements like hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, silicon. So the percentage weight of Earth's crust and human body, we are comparing it. So if you notice the percentage of weight of Earth's crust, hydrogen Earth's crust weight is 0.14 percent percentage weight, whereas human body, the hydrogen is 0.5% weight. So it is lesser than the Earth's crust. When compared to carbon, carbon Earth's crust weight is 0.03, and whereas in human body it is 18.5%. So it has, so near greater than the Earth's crust, carbon is present in the living organism. So oxygen, the Earth's crust is 46.6 percentage, whereas in human body it is 65 percentage. So nitrogen is very little. So whereas here in human body it is 3.3 percentage. Okay, the sulfur will be around 0.03, whereas in human body the percentage of sulfur is 0.3%. It is higher than the Earth's crust. So we are comparing the non-living and living matter. That is, Earth's crust is the non-living thing, and the human body is the living matter. So you can notice this differences in percentage of it. So some of the elements are found more in percentage on the Earth's crust, whereas it is lesser in the human body. The percentage weight of those components in human body or living organism is less. So you notice that nearly sodium contains 2.8%, but they are present as in very less content as micromolecules. It would be zero point 0.2. So the calcium are 3.6 percentage on Earth's crust, but in a human body it is just 1.5% of its weight. So magnesium is 2.1 percentage, whereas in human body the magnesium is 0.1 percentage. So silicon, it has 27.7 percentage in the Earth's crust. So there is very little amount of silicon. It is negligible amount of silicon is noticed in the human body. So this is the percentage weight that you notice in the living and non-living matter. For non-living matter, they have taken the example of Earth's crust, and for living matter, they have taken the example of human body. So when you compare both of them, you can understand how this hydrocarbons, they are more in living organism than in the non-living organism. So we can, it is very clearly aware about. When compared to the inorganic compounds are lesser in the human body when compared to the Earth's crust. So, like silicon, it is not at all present, or it is very, very negligible amount of weight of silicon is present in the human body. So, this sort of uh, comparison you can also do. So, the hydrocarbons are more in percentage weight in human body than on the Earth's crust. Then the inorganic components are more in the non-living thing like the Earth's crust than in the percentage weight of human body. So this comparison gives you a very clear picture of hydrocarbons being more in case of human body or living organism than in the non-living things or the Earth's crust. Okay. So the biomolecules in this, what we are going to study, if you can see the backdrop of the slide where we are, you see the RCOOH, that is carboxylic acid. You know the functional group COOH is carboxylic acid groups. So nucleoside will be there. Nucleoside is found in the DNA and RNA, that is nitrogenous base plus sugar. We call it as nucleoside. Then adenine, it is nitrogenous base. Guanine, it is nitrogenous base. Then nucleotide is nitrogenous base plus sugar plus phosphate group. We call it as nucleotides. So the DNA and RNA are polynucleotides. So you should be understanding about nucleotide is the nucleic acid is made up of this polynucleotides. Then you also notice here the amino acids. So different types of amino acids you find in the living organisms. RCHNH2, COOH. So amino group as well as carboxylic acid group is found within this amino acid. Then you find here glucose. Glucose is a carbohydrate. So you notice this type of different types of biomicromolecules within the organism. So you notice here the different types of molecules. So we are going to study here biomicromolecules. So biological micromolecules, you can analyze them by this uh, the dry ash method of it, the biological micromolecules. You find it in the filtrate, isn't it? When we did the living tissue, when we made a solution in trichloro acetic acid, we found the biomicromolecules were present in the filtrate. Biomacromolecules which had greater than 1000 Daltons was found as retentate. Okay. The biological micromolecules, they include amino acids, lipids, sugars, and nitrogen bases. So the biological micromolecules which are having less than 1000 Dalton weight and which are found dissolved in the, which are found as filtrate, they would be containing the amino acids, lipids, sugars, and nitrogen bases. So the biomicromolecules include these. So let us understand the properties of these biomicromolecules. So the molecular weight of biomicromolecules, that is micromolecules, or you can also call it as biomolecules. So they are found in the acid soluble pool ranges from 18 to 800 Dalton DA. Okay, Dalton is a unit. So that is their molecular weight. So lesser than 1000 Daltons. So their molecular weight ranges from 18 to 800 Dalton. The acid soluble pool, they represent the cytoplasmic composition. So the acid soluble pool, they represent the cytoplasmic composition. So these are all the cytoplasmic composition. So the micromolecules are all found within the cytoplasm. So that is what you have to remember. So the biomicromolecules are seen in uh, the filtrate which passes through the cheesecloth or the cotton filter. So they are the acid soluble pool. So the acid soluble generally they represent the cytoplasmic chemical composition. So the cytoplasmic components are seen in this acid soluble pool. So the acid soluble pool generally represents the cytoplasmic composition. So what are the biomicromolecules which are found, or biomolecules they also call it as? So this micromolecules are biomicromolecules which is found in the acid soluble pool or in the filtrate. They are amino acids, lipids, sugars, and nitrogen bases. So we'll try to understand the amino acids as the first component of it. So as I've already told you, they have an amino group and carboxylic acid group. So and R is a variable side chain. So the alpha carbon. So you know that carbon is tetrahedral, isn't it, in structure? So you can notice this tetrahedral structure of carbon. So the amino acid, it contains the amino group and carboxylic acid group. There is a variable side chain group, which we generally call it as R group. It is a variable group. Okay. So the amino acids are compounds formed of an amino group, NH2, and an acid group, that is the carboxylic acid group, C over H, and a variable group R. So the compounds formed of an amino group, carboxylic acid group, hydrogen, and a variable group R is called as the amino acid. So the NH2 and COOH are attached to the same carbon atom, that is the alpha carbon. So we call this carbon atom as the alpha carbon. So they are called alpha amino acids since they are attached to the same carbon atom, the carboxylic group and amino group. So to the same alpha carbon. So we call it as alpha amino acids. So they are substituted methanes. So they are the substitution of methane. CH4 has been substituted by one hydrogen has been substituted by the amino group. One hydrogen has been substituted by the carboxylic acid functional group. The other hydrogen, it is substituted by the R variable side chain. So you know that they are substituted methanes, you can call it as. So alpha amino acids are substituted methanes. Okay. So that is what you have to remember regarding the amino acids. So what are amino acids? They are compounds formed of an amino group, that is NH2, an acid group, COOH, H, and a variable group R. So the NH2 and COOH are attached to the same alpha carbon. So they are called as alpha amino acids. So you can also call this alpha amino acids as substituted methanes. So the amino group and the carboxylic group, R group and H group and R group, they are substituting the hydrogen components of methane. That is why we call this alpha amino acids as substituted methanes. Now, amino acids, you can find different types of amino acids. Like if R group has only H, okay, the variable group is H. So it is going to be CH2, COOH, NH2. So we call it as glycine. This type of amino acid is glycine. If the R group, okay, so if the R group is having the methyl group CH3, okay, if our group is having the methyl group CH3, we call it as alanine. So you can just see glycine has the R amino acid, instead the R has H. Okay, the alpha amino acid has R replaced by H, the variable group. In case of alanine, the R is CH3 group, that is the methyl group. And in case of serine, the CH3, the R group is the ethyl, methyl alcohol group or CH2OH group. In serine, they would be having the R replaced by CH2 OH group. See in the structure, you are noticing this highlighted here. So you can notice a tetrahedral carbon, isn't it? So there is a tetrahedral carbon, so which is attached to two hydrogen. The white represents the hydrogen part of it. So then the blue represents the NH2. Blue is nitrogen and white are hydrogen. So the red part of it, so the gray color is the carbon. Okay. So the red part of it is oxygen and white part is the hydrogen. So this is nothing but the COOH functional group amino group, and the R group is replaced by hydrogen, so we call it as glycine. If the R group is replaced by amino, the methyl group, that is the CH3 group, so we call it as alanine. And if this R group is replaced by CH2OH, okay, so the methyl alcohol group, we call it as serine. So we have studied the structure of glycine, alanine, and serine, which are amino acids. So very easy to remember, isn't it? These are all alpha amino acids. So where you find uh, they are the substituted methanes. Okay, the R is going to vary. In one of them, glycine, R is H. In alanine, R is going to be CH3, that is the methyl group, and in serine, R is going to be the CH2OH group, that is the methyl alcohol group. So this is how the amino acids vary according to the component of R in case of alpha amino acids. So in your organisms, 20 types of amino acids are used in protein synthesis. So they are essential amino acids. So 20 types of amino acids are used in protein synthesis. So the amino acids are of three types. The acidic amino group, okay, the acidic amino group, example glutamic acid. So where they will be having this uh, the components of this will be the OOH group. Okay, so the acidic amino acids, example is glutamic acid. The basic amino acids, example is lysine. So where they have this NH2 groups will be present. Okay, so in the acidic amino acids, the carboxylic group will be two in number. In basic amino acids, the amino group will be two in number. Example, lysine. In the neutral amino acids, you find uh, example is valine. So there would be neutral amino acids will be having one carboxylic acid group and one amino group. So they are the three types of amino acids. So 20 types of amino acids are used in protein synthesis. So you should remember all these black structures are the carbon. The red structures represent the uh, blue color is amino. The red structures represent the oxygen. Okay. So the gray color, they represent the carbon. So white color, they represent the hydrogen. So you can easily study the molecular structure of these. Okay. So you know carbons are tetrahedral, isn't it? So they fulfill this. So the amino acids are of three types. The acidic uh, amino acids, they have two carboxylic group. The example is glutamic acid. So two carboxylic acid groups they'll be having in acidic amino acids. In basic amino acids, they'll be having two amino groups. Example is lysine. Then in the neutral amino acids, you are going to notice that so they have one amino group and one carboxylic acid group. So we call them as neutral amino acids. So in uh, amino groups, acidic amino acids, they have two carboxylic group and one amino group. So in case of basic amino acids, they are going to have two amino groups and one carboxylic groups. And in case of neutral amino acids, they have one amino group, one carboxylic acid group. So this is what are the types of it. So the amino acid groups example is glutamic acid. The basic amino acids example is lysine. The neutral amino acid examples are valine. So you have got exposed to three more amino acids, that is glutamic acid, lysine, and valine. In the previous example, you have got exposed to glycine, alanine, and serine. So the amino acids where the R group are variable and they are replaced by H, CH2, and CH2OH, you have them as gas, you can remember it as, that is glycine, alanine, and serine. So here you are having this acidic acid groups, glutamic acid, GLV, you can remember there it is gas, glycine, uh, the alanine, and serine. Okay, so here you can remember it as GLH, glutamic acid, lysic acid, lysine, sorry, not lysic acid, lysine, and valine. Valine is a neutral amino acids. And all these three are also neutral amino acids because the carboxylic acid and amino group, they are single in number. So these sort of amino acids, we call it as neutral amino acids. As I told you, this chapter is more on the chemistry part of it, concerned with living organism, less of biology part and more of chemistry. So there are 20 types of amino acids which are used in protein synthesis. So we should remember it. So 20 types of amino acids are there. So we'll also be studying this in the molecular biology where we are going to discuss about the triplet codons which has that capability to synthesize more than 20 amino acids during the protein synthesis. So that is possible, and that is why the codons are triplet. They are not in unicodons or uh, bicodons, they are always triplet codons. So unitary codons or binary codons, they are not able to occur because 20 types of amino acids are required for our protein synthesis. So for that purpose of it, the codons has to be triplet so that they can synthesize more than 20 type of amino acids. So some of the amino acids are aromatic. So the amino acids are aromatic. Example, tyrosine, phenylalanine, and tryptophan. So some of the amino acids are aromatic like tyrosine, phenylalanine, and tryptophan. So they are the aromatic amino acids. Tyrosine, tryptophan, and phenylalanine. So amino acids can be classified as follows: essential amino acids, non-essential amino acids. So the essential amino acids, they cannot synthesize by the body and they should be supplied through diet. So they have to be obtained through diet. So what are the essential amino acids that are obtained through diet? Lysine, so which is nothing but the alkaline amino acids or basic amino acid. So leucine, isoleucine, tryptophan. So they are all essential amino acids which has to be obtained through our food. Lysine, leucine, isoleucine, tryptophan. So the essential amino acids, they cannot be synthesized by our body and they should be obtained through diet. So whereas in non-essential amino acids, they can be synthesized by our body. So like glycine, alanine, serine, arginine. So the glycine, alanine, serine, arginine, they are all synthesized within our body. So they are non-essential amino acids. Essential amino acids are obtained through our food diet. Okay, so the essential amino acids, some of the examples are lysine, leucine, isoleucine, tryptophan. The non-essential amino acids, they are the gas, you can again remember about them: glycine, alanine, serine, arginine. Okay, they are all the non-essential amino acids examples. So these might be asked for 2 mark questions with example. Define non-essential amino acids with examples, with any 2 examples. So you should be prepared for that sort of questions in this topic. So you should understand in amino acids, the NH2 and COOH, they have ionizable nature. So the structure of amino acid changes in solutions of different pH. So the amino acids, the amino group and carboxylic acid group, they have ionizable nature. So the structure of amino acid changes in solutions of different pH. See, now you can notice that they have this amino group is positive. So the COO minus, they are the negative part of it. So this we call it as bitter ionic form. Okay. So if both amino and COOH are ionized, okay, so it is called zwitter ion. So if both amino acid and carboxylic group are ionized, it is called as zwitter ion. Okay, so here you can notice only the amino group is ionized, and in this component it is only the carboxylic group which is ionized. If both amino and carboxylic group are ionized, we call it as zwitterion. So in amino acids, amino and carboxylic acid, they have ionizable nature. So they can exist as separate ionizable component. Only the amino group might be ionizable, or only the carboxylic group might be ionizable. But if both of them are, so the structure of amino acid, that is the reason they change in solutions of different pH. Acidic, they might have a different reaction. In a basic, they might have a different pH. If both amino and carboxylic group are ionized, we call that type of form, that type of uh, amino acids as zwitterionic form. If both amino and carboxylic group are ionized within the same amino acid, we call it as zwitterion. So you should remember about the definition of zwitterion. What is zwitterion? If both the functional group, that is amino acid, if the functional groups, the amino acid and the amino group and carboxylic group both are ionized in the same amino acid, we call it as zwitterion. So remember about this zwitterion. So they are the zwitterionic forms.
Sir, can you go to the previous slide once? Sir,
Mm. So what you have to know here, this is the slide that you require. So in this, you should just remember the essential amino acids are obtained through the food diet. The non-essential amino acids, they are synthesized within our body. So example, you can easily remember the non-essential amino acids as gas. We have already discussed this example in the where the R group is variable. Glycine has hydrogen, alanine has methyl group, serine has methyl alcohol group. Instead, the R group will be methyl alcohol. So you can easily remember this glycine, alanine, and serine as example for non-essential amino acids. Essential amino acids, you can remember leucine, isoleucine, tryptophan, lysine. So you can remember these are the examples of essential amino acids. The essential amino acids, they are synthesized. They cannot be synthesized our body, and they have to be obtained through the food diet. So the various food, fruits and vegetables, they are source of this essential amino acids. So our food should also be diversified. So that was also one of the reason for it. So you might not get all the amino acids in one source of food. So food should be diversified to obtain this different types of amino acids, which are essential amino acids that cannot be synthesized by our body, and it has to be obtained only through the food that we consume. So the various types of food that we consume, they are the source of these essential amino acids. Okay. So these essential amino acids cannot be synthesized by our body, whereas non-essential amino acids, they are synthesized by our body. Example is gas, glycine, alanine, serine, arginine. Yes, have you understood about it?
Excuse me, sir.
Yes.
Some amino acids are aromatic. What is the meaning of that, sir?
Aromatic, you know this aliphatic chain and aromatic chains, isn't it?
No,
The
sir.
aliphatic chain is a open chain compound. Aromatic chains are closed chain compounds. See, aromatic, as the name indicates, they would be having smell, the pleasant smell. So aroma would be there, aromatic compounds, and they also have this, the closed chain compounds will be associated with the amino acids. So that is what we call it as some amino acids are aromatic, like phenyl group. So they have phenyl alanine. So these components will be having a one of the benzyl group or the closed chain group will be associated with amino and the carboxylic acidic group. So these sort of amino acids, some amino acids are aromatic. One, they are when they are associated with this aromatic compounds, that is also one of the reason of it. Some amino acids are aromatic. Okay, so they have this benzyl group along with them in the amino acid. So we call them as aromatic amino acids. Tyrosine, phenylalanine, and tryptophan are examples for the aromatic amino acids. They have this benzyl group, the closed chain group. So you have those in the basic study of chemistry, isn't it? So your chemistry teachers would also be discussing these aromatic compounds, aliphatic compounds in your uh, chemistry chapters when they are doing it organic unit, organic compounds. There will be organic chemistry. Will be discussing on these concept of it, aliphatic chain, aromatic chain. So you would be knowing, and they will be discussing more about it. So they'll be associated with benzyl group. That is also one of the uh, reason for it, that the amino acids are aromatic. Understood about that?
Yes, sir.
Okay. So the next part of it that we are discussing after amino acid is lipids. See, there are various types of lipids. You have this fatty acids and oils. So the differences between fatty acids and oils, in the room temperatures, the fatty acids remain as solid, whereas in the room temperatures, the oils remain as liquid. Okay, so most of the fatty acids, they remain as solids in room temperature, whereas the oils are as liquids in the room temperature. So the lipids have fatty acids and oils are there. So the differences between fatty acids and oils will be discussing later. So the lipids are water insoluble. They cannot dissolve in water. That you have to remember. So the lipids are water insoluble. They cannot dissolve in water. So the lipids contain carbon, hydrogen, and oxygen, but number of oxygen atoms is usually less. So the lipids contain carbon, hydrogen, and oxygen, but the number of oxygen atoms are less. You have to remember about this lipids. So they are water insoluble. So the lipids contain carbon, hydrogen, and oxygen, but the number of oxygen atoms are less. Okay. The types of lipids, simple lipids. So they have given a slide with examples of it, like the coconut oil. So it's a very simple type of lipids. Compound lipids, you find it in various source of food. Then derived lipids, like in yolk of egg, you notice this derived lipids. Okay, so there are different types of lipids. The classification of lipids are of different types. Simple lipids, like in case of this coconut oil, is a very good example for simple lipids. Compound lipids are seen in various food. Then derived lipids, they show this yolk of the egg where you find this lipids with other components. So we call them as derived lipids. Okay, so lipids, they'll be composed of they are water insoluble. They contain carbon, hydrogen, and oxygen, and the number of oxygen atoms are less. That we have understood about it. So the types of lipids are simple lipids, compound lipids, and derived lipids. So they have given the slides, which are the examples for these simple lipids, compound lipids, and derived lipids. So let us understand about the simple lipids. So these are formed of fatty acids and alcohol, like glycerol. So they are formed of fatty acids and alcohol, like glycerol. The structure of glycerol and other things we'll be studying in chemistry part of it. So as I told you, you won't be having uh, the structure of they won't ask you to draw or to write the structure of this lipids, like the palmitic acid or arachidonic acid. They won't be asking you to write those structure, but you should be able to identify them. Okay, especially in NEET or in CET examination, giving the structure, they might ask you to find out what are these structures. So you should be able to identify them. So simple lipids, they are formed of fatty acids and alcohol, like glycerol. They will be having this uh, glycerol, so which has the fatty acid as well as alcohol. So fatty acids are lipids with a hydrocarbon chain, R group, ending in COOH group, that is RCOOH group. So fatty acids are lipids with a hydrocarbon chain. So they are the lipids with hydrocarbon chain. So you can notice this palmitic acid. So they are lipids with hydrocarbon chain. So they have nearly C16. Okay, and they are having and they end in a COOH group. So you see this hydrocarbon chain ending up with a COOH group. So carbon and hydrogen numbers will be more than oxygen. That is what you can remember. So they end up in the COOH group. So the hydrocarbon group is represented as R and COOH. So simple lipids, you can write it as RCOOH, where R represents the hydrocarbon chain and they end with COOH. So simple lipids, these are formed of fatty acids and alcohol, okay, like glycerol. So fatty acids are lipids with hydrocarbon chain. So you have the COOH hydrocarbon chain ending up with the COOH group. Example is palmitic acid obtained in palm oil. It has 10, 16 carbons. Count them. So they have totally 16 carbons, CH3, CH214, COOH, or you can write it as C15H31 COOH. So they have 16 carbons, including the carbon in the COOH. Okay, they have 16 carbons. So their formula is CH3, CH214, COOH. Or you can also call it as C15H31 COOH. That is the formula for palmitic acid, which is seen in the palm oil. Then the arachidonic acid, they have 20 carbons. Okay, so you can notice this arachidonic acid, which have 20 carbons. So they also have one CH3 group and COOH, they have shown it. Okay, so you can separate the CH3 and COOH, or you can write it totally as C20, then the hydrogen and COOH group, you can write it as. Okay, so this is the two examples for simple lipids. Now the types of fatty acids. So we know the fatty acids have the COH group, and the OH group is found in glycerol. Okay, so the saturated fatty acids and unsaturated fatty acids. So the saturated fatty acids, they have no double or triple bond between the carbon atoms. Example is palmitic acid, stearic acid, C17H35 COOH. So they are all having no double or triple bonds between carbon atoms. The hydrocarbons, they don't have any double or triple bond between carbon atoms. Unsaturated fatty acids, they have one or more C double bond. So double bond will be there. So double or triple bonds will be there. One or more C double bond C, or bonds will be noticed in the hydrocarbons. Example is oleic acid, C17H33 COOH. It is oleic acid. Arachidonic acid, C19H31 COOH. Okay, so these are all examples of where the carbon has one or more double or triple bonds. So we call them as unsaturated fatty acids. Example is oleic acid. You can notice here there is a double bond carbon. Okay, so this is mono unsaturated fatty acid. So there is only one uh, unsaturated part where they have the double bond. That is the reason we call it as mono unsaturated fatty acids. So there are saturated fatty acids where they do not have any double or triple bonds between the hydrocarbons. In unsaturated fatty acids, they have one or more double or triple bonds between the carbon atoms. So this is noticed in case of unsaturated fatty acids. You can take example as oleic acid, arachidonic acid. So the saturated fatty acids example is palmitic acid, stearic acid. okay, where they don't have one or more double or triple bond between the carbon atoms. They do not have any double or triple bond between the carbon atoms. Unsaturated fatty acids, they do have one or more double or triple bonds. Example, oleic acid and arachidonic acid. The structure of glycerol, I told you already, the glycerol has the hydroxyl group. So the structure of glycerol, if you notice, it is trihydroxy propane. So the scientific name of glycerol is trihydroxy propane. Glycerol is nothing but trihydroxy. There are three hydroxy group, isn't it? And C3 propane group it is. Trihydroxy propane structure of glycerol. They have this OH group. So glycerol is trihydroxy propane. So they might ask you what is the structure CH2OH, CHOH, CH2OH. So trihydroxy, first of all, you notice it is very easy to remember by scientific names, IUPAC names, that is trihydroxy, and you notice this propane. So trihydroxy propane is nothing but glycerol. So you have to remember the structure of glycerol. Okay. So now the fatty acids are esterified with glycerol through ester bonds, forming monoglycerides, diglycerides, and triglycerides. So the fatty acids are esterified with glycerol through the ester bonds. So the bonds, see the glycerol, that is the trihydroxypropane, one of the hydroxy group of this glycerol and that of the carboxylic group. So they are going to combine together to form water and they are liberated. And this results in the formation of an ester bond. So forming this ester bond leads to the formation of, that is COO, you can notice here, the ester bond leads to the formation of monoglycerides, diglycerides, and triglycerides. So in glycerol, one fatty acid is attached to it. So as in this above case, so the palmitic acid, one fatty acid attached to one OH group of the glycerol, we call it as monoglyceride. One glycerol having two fatty acids attached at the OH group, we call it as diglyceride. You can notice here. So uh, the glycerol, one glycerol OH is attached with the fatty acid, so we call it as monoglyceride. If two OH groups in the glycerol is attached with fatty acids, if two fatty acids are found in one glycerol, we call it as diglyceride. If three fatty acids are found attached with glycerol, we call it as triglycerides. So you have monoglyceride, diglyceride, triglyceride, which is formed by the ester bond between the glycerol and fatty acids. Fatty acids are esterified with glycerol through this ester bond, forming monoglycerides, diglycerides, and triglycerides. So monoglycerides, they have one glycerol and one fatty acid. Diglycerides, they have one glycerol and two fatty acids. Triglycerides, they have three fatty acids and one glycerol and three fatty acids. So this is how uh, the different types of glycerides are noticed: monoglyceride, diglyceride, and triglycerides by the combination of glycerol and fatty acids. So triglycerides. So here, based on melting point, lipids or the triglycerides are two types: fats and oil. So if their melting point is very high, we call them as fat. If their melting point is very low, we call it as oils. So the fat and oils, they differ based on their melting points. So based on melting points, the lipids, that is the triglycerides, are of two types of fats and oils. So they are triglycerides, that is one glycerol with three fatty acids, we call them as triglycerides. So here R1, R2, and R3 are the triglycerides in this particular component. Okay, so here you notice that there are three fatty acids are there, so which are attached to a glycerol. So we call it as triglycerides. So the based on melting points, the lipids or triglycerides are of two types. They are the fats and the oils. If they require higher melting point, they are fats. If they are require a lower melting point, they have lower melting point, we call them as oils. So based on their melting points, so we have two types of lipids or triglycerides, fats and oils. Fats have higher melting point, oils have lower melting point. Generally in the room temperature, the oils are as liquid, the fats are as solids. So you might notice the sometimes the melting point is a little higher in coconut oil. You notice that during the winter season they would have solidified, but in summer they would be in the liquid form. But they are again oil. Their melting point is lower to that of, even then they are lower than that of fats, but higher to other type of oils that you can remember regarding the coconut oil. So we studied about simple lipids. Now the second variety that we are studying is compound lipids. So compound lipids, these are the esters of fatty acids and alcohol with additional groups. Like you can find phosphate group are found along with fatty acids and glycerol, you find phosphate. So phospholipids, when we are saying biphospholipid layer, we say for this fluid mosaic model of plasma membrane. So you should remember the phospholipids, they have fatty acids, glycerol and phosphate group. So they are found in the cell membranes. Example, the lecithin phospholipids. Example is lecithin is found in the cell membrane. So biphospholipid layer, you find it in case of the fluid mosaic model of plasma membrane or cell membrane, isn't it? We had discussed there. So what are phospholipids made up of? They are having fatty acids, glycerol, and they have this phosphate group. So the compound lipids, these are the esters of fatty acids and alcohol with additional groups. So whenever you are defining compound lipids, you should say it as these are the esters of fatty acids and alcohol with additional groups. Example is phospholipids. They have fatty acids, glycerol, and phosphate, and they are found in the cell membrane. Example for this is lecithin. Okay, so you find the lecithin here. They have the. It is very easy to remember, even though the structure looks a little complicated. So they are the esters of fatty acid and alcohol with additional group. So you have a phosphate group here. So can you notice this phosphate group? So they are attached along with alcohol and carboxylic group. Okay, so they are first of all fatty acids and glycerol plus phosphate group. So together we call them as the phospholipids. Example is lecithin. So you can notice here the uh, three part of them, the glycerol triglyceride, where you notice that one of the glycerides will be having this phosphate group. Okay, so the phosphate group, so they are found in cell membranes. Example is lecithin. You can notice these things. So derived lipids, these are the products of hydrolysis of simple lipids and compound lipids. So hydrolysis of simple lipids and compound lipids, you are going to get the derived lipids. So derived lipids definition is very simple. It is the hydrolysis of simple lipids and compound lipids. What is compound lipids? These are the esters of fatty acids, glycerol, and glycerol with additional groups. That is what is compound lipids. Derived lipids, these are the product of hydrolysis of simple lipids and compound lipids. Example is cholesterol is a very good example for this derived lipids. That is the reason they have focused it on yolk, which has this cholesterol. Okay, so the cholesterol structure of it you can notice. So the products of hydrolysis of simple lipids and compound lipids, we call it as derived lipids. Example is cholesterol. So the structure of the cholesterol you can notice here. Okay, so that would be sufficient for today's discussion. So here you can notice the four chain benzyl groups attached with that. So you have this three hexane group with one pentyl group. Okay, and they have one of this has OH attached to it. This is the structure of cholesterol. So look into the diagram and you should be able to identify, yes, this is the structure of cholesterol. So three benzyl group with one pentane or pentyl group, you can notice here. One end they have this OH group, the other end is an open chain. So this is what is the structure of cholesterol. Okay, so yolk of egg has cholesterol. Okay, that is the reason they ask us to restrict the consumption of egg to lesser numbers, even though they are a rich and cheaper source of nutrients, the egg. But the yolk, since they have more of cholesterol, so the consumption of egg should be restricted to two or three in a day. That is what most of the scientists talk about. Okay. So the next biomicromolecule sugar will be studying in the next class. So in today's class, we discussed about the amino acids, the proteins we discussed, especially the amino acids and lipids we have discussed in today's topic. So the types of amino acids, then we discussed about the types of lipids, like we had studied about three types of lipids, isn't it? Simple lipid, compound lipid, and derived lipids. Okay, so in the next class, we'll be discussing about the carbohydrate. If you have any doubts or clarification, students, you can ask me the questions or queries.